Thank you for joining the Once Changing the World, India's first future tech meets sustainability podcast. And today I'm delighted and honored to have with me Dr. Jun Wu, who's an assistant professor at the Department of Molecular Biology at UT Southwestern Medical Center. His lab mainly focuses on using stem cell models and interspecies chimeras to gain novel insights into mammalian development and evolution, as well as develop regenerative medicine applications. Dr. Wu received a Ph.D. from the University of Tennessee and completed postdoctoral training at the University of Southern California with Dr. Martin Pera and at the Salk Institute with Dr. Juan Carlos Izpisua Belmonte. Professor, really appreciate you taking time being part of the podcast. The topic of discussion today is going to be interspecies chimeras and regenerative medicines. So I think it'll be great if you can start with explaining what are chimeras. How do you develop these interspecies chimeras?
Sure. First of all, really appreciate the opportunity, Eddie, and it's great to be here. So to answer your first question, interspecies chimeras by definition is an organism that containing cells from more than one species. So we can generate interspecies chimera through several different methods. One of the methods that we're particularly interested in is to use pluripotent stem cells to inject these cells from one species into an embryo from another species, and then perform embryo transfer to let the cells from two different species to communicate with each other to generate the organism, as I said, containing cells from two different species in this particular case.
What's the longest duration of a living chimeric organism? And does this mean that, if you can take like a cell from one species and kind of transfer it to another species and develop another complete interspecies chimeras, does it mean that theoretically we can create full-blown, not just these living organisms, but possibly chimeric species?
Yes, very interesting question. So with regard to how long the interspecies chimera can live, at least in our experiences between rodents, such as rat and mice, we have been able to generate chimeras that can live the entire lifespan of a mouse, for example, between 2 to 3 years. And also there are reports that in nature, there's chimeras between species that are naturally can occur in nature, and then that may live longer, but I don't have any information regarding that. Yes, and then your comments on since we can generate these interspecies chimeras and they can be born, then potentially we can use this interspecies chimera to generate tissue and organs for regenerative purposes.
When you're talking about these full-blown chimeric, species of sorts, what's your experiments been so far? Is it being restricted to mouse, or is it going to go a step further? And what are the ethical implications of kind of doing something like this? When you're kind of tinkering around with these different cells and creating a complete new species of sorts.
So the chimeras that are born are mostly limited to rodent chimeras, although using different methods such as grafting cells, progenitor cells from one species to another, people have been using this method to generate birds chimeras like quail and duck in the past. With regard to pluripotent stem cells, those chimeras are only limited to rodent between mice and rat. And with regard to other species, including humans and pigs, the extent of chimera contribution is much lower compared to rodent chimeras, because the evolution, they were more distant from each other. And so therefore the study of those are very limited. So there are some reports on using monkey pluripotent stem cells to generate monkey pig chimeras that are born, but the chimera contribution from the monkey pluripotent stem cells is very limited. And with regard to human, there hasn't been any report that using human pluripotent stem cells to generate a born interspecies chimera in any species to date. And I think part of it is that the contribution is too low. There's just no, at this stage, and it's not practical. And also ethically, we need to evaluate several different parameters before we can allow these chimeras to be born.
What's the purpose of creating these interspecies chimeras? What could be the useful applications of doing a work like this?
Oh, there are many different potentially interesting questions that we can ask. for example, from a developmental, and a fundamental biology point of view, I think this is a fascinating system to have cells that evolve separately for millions of years in the same in vivo system, so that we can study their crosstalk with each other's, in hope that we can understand evolution and also understand development, in a different way. For example, there are several questions that arise during the generation of interspecies chimeras, that the organ size and body size, how those are controlled, because chimeras such as mice and rat, between mice and rat, we know that the rat is much bigger than mice. But surprisingly, when we inject the rat pluripotent stem cells into a mouse embryo and generate a rat mouse chimera, the body size and organ size is exactly the same as a mice. So that suggests that the host environment is critical in determining the organ and body size. And this platform can potentially give us a insight into how organ and body size are controlled. And then the second question is developmental time and pace. We know that different mammals, different animals develop at different rates, and gestation period varies among mammals. And we found that just like the body and organ size, the developmental time is more similar to the host species, suggesting this is a non-cell autonomous control of developmental pace and time during development, which is quite interesting. I think this interspecies chimeras can be used to understand the mechanism underlying these. And recently, we realized there is a very intriguingly, the transcriptome, the gene expression of the donor cells are maintained the same as the donor species identity, suggesting that there is a cell intrinsic mechanism in the interspecies chimera as well. So even though, for example, the red cells growing inside the mouse embryo, they still maintain largely the red transcriptome, which is fascinating. I think the interspecies chimera can be used to study the crosstalk between the microenvironment and the genome, how they shape during the process of the development. But most importantly, I think probably the focus of our conversation today is the interspecies chimera can be potentially used to generate organs from one species inside another species. This is very relevant to humans because there is a worldwide shortages of organ donors, and researchers like us and many others are scratching our head trying to figure out a way to generate organs that can provide supplies and then overcome these shortages. And I think that using interspecies chimeras, and particularly using a strategy called interspecies process complementation, can potentially give us, these human organs, generate human organs in the animal species.
How cool would that be? Because you rightfully pointed out, there is a global shortage, when it comes to organs. I mean, globally, in India also, it's like a huge problem. So I had interviewed Dr. Mike Curtis, the CEO at eGenesis, and they are developing. They're genetically engineering pigs that can host, these biocompatible organs. And you had recently written this paper titled "Towards Developing Human Organs via Embryo Models and Chimeras." I mean, can you kind of elaborate on this entire work on growing human organs in animal hosts?
Yes, as you said, there are several different strategies, very promising strategies to overcome this worldwide shortage of organ donors. And one particular one is a xenotransplantation, which is really advancing fast in recent years. These success recently are built upon like decades of work to understand the barriers, the immune barriers between pigs and humans. Because in the earlier days, in the late 90s and also early 2000, I think researchers try to transplant pig organs into humans, but failed because of immune rejection. And then with the progress on the gene editing and understanding the immune system better, then we have the strategy now to knock out certain genes and knock in certain genes that can make a human and pig immune system more compatible with each other. That result in the recent success in transplanting human organs in several patients, terminal AO patients. And I do think this strategy will be translated into the clinic very soon. But one of the issue with this strategy in the long term, right, is that we don't know whether these are 100% pig organs and pig cells. I mean, some organs, it works pretty well because many organs like heart they need more like mechanical functions rather than metabolic functions. And then I think for organs like a liver and the lung, maybe metabolic functions are more important. And there, in those scenarios, you will need human cells to do the job, right? So I think that's why we believe that this interspecies chimera approach will be the next step. So on top of these genetically modified pig that can reduce the immune barrier, if we can enrich these organs with human cells, then I think we will provide a better strategy in the future to overcome the organ donor shortages.
So, Professor, you're saying that maybe eGenesis and others who are taking with the genetic editing approach by knocking off and maybe adding a couple of genes might not be the right pathway for human compatible organs. Can you kind of then elaborate your approach of these interspecies chimera that you're talking about? Where does the research stand at this point in time? And when do you think that we will be seeing this moving from the research to the healthcare industry?
Yes. So interspecies chimeras are random. A contribution often random. It's really hard to control. So one of the strategy that I briefly mentioned is called interspecies process complementation. So the concept is very simple. It's instead of using a wild type host embryos for chimera generation, now we can use genetically modified host embryos. So modified in a way that a certain organ will not form in the host species. For example, if you knock out a gene called PDX1, the host embryo will now generate a pancreas. And then with the donor cells, because it is not modified, it still have a perfectly functioning copy of the PDX1 gene, then they can take advantage of the situation and generate a pancreas completely from a donor proponent stem cells. So this has been successful between rodents, specifically between mice and rat. So we can generate rat pancreas inside a mouse embryos and the other way around, a mouse pancreas inside the rat. And also, in the past decades, researchers have been trying to target other organs like kidney. And recently, we also had a success with the brain tissues, although this will not be applied to humans in the future, but it will be a good fundamental tool to study brain evolution among rodent species. But with regard to other species, specifically human, the progress is still very limited. Although more recently, there's report from China. One of the friends of mine, they were able to generate metanephros, human metanephros in the pig, basically the precursor for the kidney cells. I think the result looks very impressive, even though it's very at the early stages. It's about 4 weeks to 5 weeks of pig development. They were able to enrich the pig kidney with 60 to 80% of the human cells. I think this, although as I mentioned, between human and animal chimera and organogenesis is still lagging behind the rodent studies, but it's inching forward and then making progress, exciting progress day by day.
Right. So, Professor, what do you think is going to accelerate this and make it move from the lab to the market? Because right now you're saying the experiments are largely between mice and rats, and the pancreas. I mean, so what would it take to scale it up from moving from mice to rodent models to maybe humans? And you mentioned about these brain tissues. So you. Can you kind of also elaborate on that? Do you mean that, you take brain tissues of a mice and kind of add it to a rat? How does that work? Will be great.
Okay, yeah, I'll answer your second question first. So in this particular experiment, we knock out a gene in mouse embryos called HES-X1. So lacking this gene, the mouse will not develop a forebrain. And then we use rat proponent stem cells inject into the mouse embryos that deficient of this gene. So the rat cells can occupy, basically generate a forebrain tissue inside the mouse. So you ended up with a chimera that the forebrain of the chimera is coming mostly from the rat cells. And the chimera still look like mice because the body size is exactly the host size, and developmental pace is exactly the mouse pace, but they have forebrain tissue that came from the donor rat proponent stem cells. And that opened up the door to understand how the behavior, how all the other associated with function associated with forebrain can vary between these two species. And we are still very at the early days to look at these behavioral tests because they're there is not a good behavioral test to distinguish mice and rat at this currently. So we need to develop other tests to understand better.
What would be the implications, the neurological diseases with something like this?
Oh, yeah, they may have some relevance to neurological disease because one of the potential application of using human proponent stem cell iPS cell derived neurons is to transplant these neurons into a host, right? I mean, it's not exactly the same as the interspecies, but still deal with the same issue as transplanting in vitro cultured, cells, which is different from in vivo developing neurons, and how that environment and the donor cells can crosstalk. I think some of the information we learned from rodent brain chimeras can be useful to help to address some of the barriers that potentially involve in the transplanting human neurons into humans in the future. But as I said, it's not for interspecies human brain generation. Definitely, that's not where we're going. But I think I want to come back to the first question, which I think is more important, is that the reason why there is such a limited progress with the human-animal chimera is because the chimeric contribution of the human cells is very limited in the host embryos. And part of the reason is that the host embryos, the host species that we can have access to are very different from humans. For example, mice and pig. Even though pig is more similar to human, but still they are separated from human about 100 million years ago almost. But between mice and rat, they are more closely related between 16 million to 20 million years, separating evolution. So as a consequence of that, I think there is a lot of differences during early development between human and pig and human mice. And my lab is dedicated to understand these differences and what we call xenogenic barriers. Basically, the mechanism that preventing the human cells from efficiently contributing to the chimera formation in those species early on. And through developing system in vitro, we in the past few years, we were able to identify at least two major barriers. One of them is cell-cell competition, which is very interesting in itself, is that we found that the proponent stem cells, for example, from mice, can out-compete human proponent stem cells in the dish. So they actively go after human cells, and the human cells are targeted for elimination through apoptosis. And then through building this in vitro system, we understand that the ancient innate immune response were involved in this cell-cell competition. And by blocking this pathway, for example, the F-kappa B toll-like receptor related pathway, we were able to prevent the human cell from dying when co-cultured with the mouse proponent stem cells. And then we translate that also in vivo. We observed that by manipulating this pathway, we can improve human cell chimeric contribution in the mice. And more recently, we have a paper coming out next week that we identify another barrier, which is a cell-cell adhesion incompatibilities because we know that even during the development of the same species, cell-cell adhesion is very important for separating of the early progenitor cells into different tissues and organs. So it's very important for cell sorting and also organogenesis. But between species, as you can imagine, due to genomic evolution, those cell adhesion molecules are incompatible between species. So we develop a strategy using synthetic biology approach. We can express, for example, nanobody and antigen on the cell surface from the two species. Then using this artificial glue to kind of promote the adhesion of this between cells from two different species. And through this strategy, again, we improve the chimeric contribution of the human cells in a host, in this case, the mouse embryos. But those two barriers are not the only barriers. As I mentioned previously, there are other barriers. There is a ligand receptor incompatibility potentially, and there is a different cell cycle and differentiation speed. They're likely serving as another barrier, and maybe there are other barriers as well. So I think in order to match human animal studies to the success of the rodents, we need to overcome these barriers. And by doing that, then we can scale up the production of the human organs and tissues in host recipient species such as a pig. It's a long way to go, but I'm very optimistic. By understanding the fundamental biology, and then that can help us to develop strategy to make this approach or method more effective in the future.
Who are the interesting researchers around the world who you looked up to or are inspired by or have a similar approach of what you're doing with the work that
Yes,
you're doing?
so I'm this whole project or my research direction is inspired by Hiro Nakauchi. He pioneered the process complementation approach. He published the first paper in 2010 in Cell that demonstrate this strategy, right? Using this process competition to generate pancreas from the rat pluripotent stem cell inside the mouse. So that he inspired me. At the time, I was still a postdoc at the Salk Institute. I saw that paper and said, "Wow, this is great." With my expertise, my contribution to the field is that because I came from a pluripotent stem cell background, so and especially human, we take this rodent studies into to the study human pigs. We generate the first human pig chimeric embryos, and also human monkey chimeric embryos while I was leaving Salk and starting my own lab. But through this process, we realized the chimeric contribution is low, is not enough. That's why when I start my lab, my main goal of the lab is to understand why this is low. What are the mechanisms? Can we develop strategy to overcome this low chimerism? This is exactly what we did in the past 5 to 6 years. And I don't think anybody in any other labs are as dedicated or as focused as our lab to try to understand these fundamental questions. But there are other labs that are trying to use whatever we found between human mice and other species, try to apply to human pig, including the researchers that I just mentioned from China. They take advantage of the information that we learned that the cell competition play a role, and also different culture condition of pluripotent stem cells. That led to the milestone study that they published last year on generating human metanephros in the pig. But I think with regard to fundamental mechanism between interspecies chimeras, we are the only one in the lab in the world, I believe, that are keep working on it.
Wish you the very best and everyone at the lab. Professor, does your work intersect with de-extinction? Because there's professors such as George Church with his startup Colossal is trying to kind of bring back the Tasmanian tiger and the woolly mammoth. Does your work intersect over there?
Yes, we actually, since you mentioned, the Colossal is right across the street from our institute. So I'm actually a advisor on their scientific advisor. So because we also work with pluripotent stem cell from other species, I think interspecies chimeras can be approached to generate gametes from endangered species or extinct species if we can get cells from those species. And then one of the work we are currently undertaking is to use, for example, northern white rhino iPS cells, and then to generate chimeras in the mice and rat, or more closely related species such as horse, to generate the early progenitor from those primordial germ cells from those iPS cells. So I think our work is just another potential application is to help species preservation and hopefully can help revive some of the already species that are endangered, critically endangered, already extinct species. But we're not there. This is just the hope, but we're not there yet.
Right. So biotech, it's there everywhere, from CRISPR, genetic editing, and you were with at the Salk Institute with Dr. Juan Carlos, who's at the forefront of age reversal with Altus Labs now. What are your thoughts on CRISPR-Cas9, genetic editing, and age reversals, and tech stacks such as that? When do you think that all of these are going to be accessible to everyone? Do you think is it possible where where we could have age reversals where people humans in the age of 80 could just possibly maybe pause or actually reverse aging?
Oh yes, very good question. I think many people may hold different views. So what I'm going to tell you is just my view only. I believe the recent excitement of all of these technologies are fueled because we're in a very exciting era that has a technical explosion. Basically, there is CRISPR, there is iPS, there's many other, cryo-EM, many other technology, including AI, right? So we're in the era that we see the explosive growth of technologies. So the consequences of that are they generate excitement. People are wondering in their, imaginations that these can solve all the problems that we have. But my view on this is that they are still far away. I think we still need to do the focus on the fundamental basic science. I think all these effort is great in the way that they can, even though we may not get there what the original proposal are, but through the process, we can learn so much, and then we can develop so much new technologies that can take us there closer. Just like give you an example. Back in the 60s, the United States and the sent men to the moon. At the time, it's a really noble goal. The outcome is a success. But I think the more importantly is during the process of making the effort, the grandeur effort, we develop technology. Now we see everywhere, like all these different industries, communications and travel, things like that. So I think it ultimately will benefit humankind, even though we start with a goal that seems unachievable at this moment, but still there is a lot of values of pursuing that dream. Yeah.
Professor, you were mentioning that, there is a convergence sort, and, that AI is kind of intersecting with biotech. How do you think artificial intelligence is going to play a role in possibly understanding biology better and maybe possibly in then creating, better healthcare solutions?
I think that's a future. I do think the AI is very helpful in helping us to filtering through all this data set, right? We have omics data set from everything that you can imagine. I think as a human brain, we're not very good at processing this complicated data set. I think with the AI's help, that can give us free us from this burdens of analyzing this tedious data set, and came up with a hypothesis based on what AI's output and then what we can learn from them. So for me, I think it's great. And also the other benefit of AI is that they can take over many of the the work that is repetitive and also need a lot of we don't need really a brain power to do these works. So I think that will be very helpful. And but again, there is many things that we don't understand about AI, that we're concerned about AI. I think in the next few years there will be better understanding. Hopefully there will be some reassurance that
Right. What are your thoughts on the entire regenerative medicine field? What are the other applications maybe you can point out to you, which you think there's going to be possibly near term and long term, which is going to be possible?
So regenerative medicine, it's in an exciting time right now because after almost 2 decades of development and testing, and then preclinical trial, I think now we are starting to see some products are entering into the clinical trial stages, including the beta cells and RPEs developed from iPS cells or the pluripotent embryonic stem cells. I think in the next few years and decades, these going to become more and more prevalent and popular. We'll see some products in the clinics can be applied to patients. But with regard to organs, as I mentioned, the xenotransplantation will see its application in the preclinical trial or clinical trial, or even in the clinic in the 5 to 10 years on some organs like the heart, I believe, and potentially kidney. That's people seeing the most success of these 2 organs in the pig to monkey transplantation in the past. But then there are other strategies like in vivo CRISPR editing. I think that can, for now, they are focusing on the cancer, but I think these concepts can be used for regeneration purposes and for the other strategies to improve the transplantation success. These are the 3 areas that I see in the near term. But in the long term, I think we still need including our approach in generating organs in animals that are composed mostly of human cells, and even using binding engineer approach to 3D printing organs, and also from other strategies, I think, or stimulate in vivo regeneration without performing this invasive surgical procedures. I think that will be the next thing to be focusing on. It's just my view. Yeah.
Is there a specific challenge that if you solve could accelerate interspecies chimeric research?
Yes, the challenge I think we're working very hard on is the stochastic nature of the chimera formation. So even though I mentioned that the process competition can help you to enrich in the organ that the donor cells, right? But outside the organ, target organ, the chimera contribution is still random. So I think that it's challenging to target the donor cells into specifically that organ. I think that's what we're working on right now. Although I don't know whether it's possible or not, but there's some strategy that we can think to make it happen. But I think that will be the challenge, to control the donor cells once injecting the embryo, where they go, what tissue they contribute. That will be the challenge. If we can achieve that, I think that will be fantastic. That can open up not only for human organ generation, but for developmental biology, right? Because you can now using donor cells that can target to a specific cell type, and you can modify them in vitro in a specific way to understand this interaction between modification and the environment during development. Yeah.
Right. Professor, my last question to you. Would you be able to kind of like paint a picture of what the world is going to look like, when all of these kind of is accessible to everyone? So possibly like maybe what I'm trying to say is maybe like paint a picture of the future of healthcare, how the healthcare is going to look like in the next possible future that you imagine.
Oh, yeah, this is my favorite question. So I'm a big fan of science fiction. I read a lot of science fiction books, and I see that, if everything that I said, everything we did discuss about can become a reality, I think you can imagine a world that you can replace your organs and tissues at will, because we know that during the aging process, it's not or in many of the cases, it's just a one organ failure. You can, just like a car, if an old car, if it breaks down, if you replace the engine, you can run it for another 20 years maybe. So I think we can see that coming potentially, either through the organ generating the pig or some other species, or the stimulation of organ regeneration in vivo, right? That's one of the things. But I think, during this period, there will be other technologies that are potentially can blow our mind and, lead us to a different direction, especially with the neuron, the machine and the brain interface, and also AI. You never know. There might be a world that we don't need a physical appearances, and we can just upload everything into a cloud or things like that. But that's just a pure imagination at this stage. But that's tell me that our imagination never stops, right? I think that's the beauty of science, the beauty of you guys, the newer generations. I think just don't limit yourself and just think outside the box. You never know. Some technologies maybe in the audience today, they'll be already growing in your brain, and then in the next 10 to 20 years, you will make it happen and just blow everybody's mind. So I that's why I'm in science in the first place. Is that just, and there's a lot of unknown and there is a lot of unpredictability, but there's certain predictability as well. But with regard to the future, the technology now we're excited about may not be the ones that are bringing us there. But certainly it helps, helps to people think about the next step.
Professor, really, really appreciate you taking time and being part of the podcast. Wish you and the team the very best. I guess just science is all about that, because we humans are constantly trying to kind of create things. Obviously, when you create things, there are knowns and there are unknowns. With the unknowns comes those risks, but also comes opportunities, because there is so many people in the world who are like, waiting for organs. And I mean, the world would be so beautiful if we could be able to solve the organ shortage problem through these interspecies chimeras. So wish you and your team the very best. And to my listeners, if you like what you see in here, then please press the subscribe button. Until next time, see you guys. Thank you. Thank you, Professor. Really, really appreciate this.
Thank you for the opportunity again. Thank you.
Thank you.